Hi, everybody. Thank you for choosing the best breakout session of the day. You can give yourself an applause. Oh, now at the end. All right, so the topic of this session, and hopefully it'll be a lively discussion because this discussion is really all about community and something that I think the HCA is doing pretty well at, I think. And then we can see here in this meeting, things really come together after a couple of years of efforts, and the various atlases are materializing, and really fascinating new biology comes out, as we just saw in the keynote. So yeah, so just some announcement formalities at the beginning. Everybody should know that this session is being broadcasted and recorded, and we posted on the event website by the end of it. So by being in this room, you are consenting that such recording and so on. And that also goes to the folks on Zoom. I know that we have a few, we have about 20 participants now on Zoom together with us. People on Zoom, you are more than welcome to join the discussion as well. Just send a chat that you want to talk, and then just unmute yourself, and then you can express yourself as well. People in the audience, we have these. So just talk to the mic so people can hear you. All right, that's the administrations. Now we can start. So just by ways of very quick introduction. So my name is Nir Yosef. We have Marie here, my colleague Mariana Boroni. I'll just make some quick introduction, and then Marie will give her introductions, and we'll start the discussion. So the way that I put it here in the title is what the community can do for me, in a sense. How can the community facilitate and accelerate progress? And we divided it into two parts here. On the left, we have resources, which are data, tools, which can be computational tools, and knowledge. And on the right, we have means that we can actually do something with those resources by sharing, benchmarking, or consolidating. And I hope that by this matrix or connection between these two components, we can have some discussion here. So I'll give some examples. What do I mean by that? So for example, data sharing or data benchmarking. By data benchmarking, I mean in terms of quality of different data sets, and see which one is good and which one I can rely on more or less. Right. So we have all these atlases that take multiple studies together. We've seen some of these today. So that's an example for an effort that facilitates that. In terms of tool sharing and consolidating, right? So all the open source environment that is out there. Also in terms of consolidating, this is pipelines that people are putting together, best practices, and so on. That also takes me to benchmarking. Malte gave his talk today about skip metrics and many other efforts that are out there. The open problems in Kaggle, just all these efforts that we can see which tools are doing well and how to make decisions. And finally, in terms of consolidating, right, how to take tools and knowledge that we have from using these tools and consolidating in terms of best practices and just really guides that are very rigorous, that go throughout an entire application pipeline rather than a particular study. So that's another area that is being evolving, right? So these are just a few examples for how do we go between the resources and the community mechanisms, if you will. But of course, there are many challenges ahead. Now let Marie.
Thank you, Nir. So as Nir said, we are seeing an explosion in terms of production of data, also in terms of diversity of backgrounds, of number of tools, and many of them kind of somehow overlap between each other. So the last census I got, I mean, a paper that described it, we have so far more than 1,400 tools available. So, and many of them, they are not very user-friendly. So for newcomers, especially the ones that doesn't have bioinformatics skills, this sometimes can pose a challenge. And also in terms of advance, we are seeing a spectacular movement towards data production, but the costs associated with data generation, they really haven't decreased so far. So for people from developing countries, for them sometimes it's very hard to produce data and to be competitive in terms of, you know, generating big data sets to analyze. So we are here today to really hear you all and to think together how we can address those issues, those challenges, and as a community, how we can become, you know, more accessible, how we can increase the availability of tools, data, and so on. So to warm up a little bit, to break the ice, I would like you guys to just a moment, to scan this code. So this is going to direct you to a web page where you can answer this question. Also, people online, please, if you can help answering this question. So what do you think it is the main obstacles preventing the wider availability of data access and analysis in our community? And while you are all answering this question, the idea was that I could show you a word of, you know, a cloud word here, but because we have some small technical problems, you guys have to believe me, I'm gonna read what we are gonna see. Let me see if we have read some responses.
People on Zoom, could you please mute yourself unless you want to present a question?
Okay, so we have the word, the cloud start to forming. So the most regular word was data so far. We also had problems with funding, formatting, metadata, standardized data, availability, lack of training. Let's give one more minute, because you guys, you can answer many times as you want. So you think there are more than one problem, you can also answer again. So, okay, the cloud's better now. So we are seeing recurrent words are funding, lack of metadata, data, training, also scalable protocols. Oh, this is getting interesting. Okay, so think on that. Yeah, as you probably can see very well, but yeah, most of the words that are very recurrent here, they're funding and lack of data, metadata. So think on that. Would you guys elaborate on that to see how we can address this challenge?
Who wants to start? Giving some more context to the model here. So, for example, we heard about metadata, we heard about funding, we heard about training. Anybody wants to elaborate on that? No? Okay. Yes. All right.
Well, related. Good morning, everybody. My name is Vinicius Maracaja Coutinho. I'm Brazilian, but I'm currently at the University of Chile. And I would like to put on discussion about the gap we have in infrastructure. For example, something that we always have when we are working with people from the International Society for Computational Biology, we have some discussions about the lack of infrastructure for training. And I'd like to hear your opinions. From our side, we are trying to develop a lot of notebooks using Google Colab that work perfectly to train people in Brazil and in Chile. But I would like to hear from you guys what your opinion about that, on how to manage this kind of issue.
Who wants to elaborate on the answer?
So when we think about the limited resources, right? So that can come from the training programs, or it can come from the actual resources like the compute equipment. So which one you think is more severe? Because you're trying to think here about broadly. So yeah.
Yes. What do you think it's going to be severe part?
No, it's because what we see we, together with people from the ISCB, we launched it 2 years ago, the Latin American Caribbean Forum on Education in Bioinformatics. And we invited people who are not bioinformaticians to be together with us discussing, because we all are we're bioinformaticians, but a lot of people is not. People behind medicine course, people behind biological science course. And one of the issues that they always raise is because they don't have computers to give classes on bioinformatics, for example. And maybe a solution could be using these notebooks. We started with a Jupyter notebook, then we realized that we still need to have an infrastructure to install everything, or you need to have a little bit of money to pay a cloud for that. But last year we started to work on Google Colab, and it worked perfectly for us. We are generating a lot of materials that we'll be using Welcome Connecting sites that we'll have in Rio in the end of this year. And maybe we'll be using also at the Human Atlas Latin American workshop that we have in Santiago in Chile. And this is maybe one way to try to solve this kind of problem in order to train more people, not only bioinformaticians, but people who want to start a little bit on that.
Thank you. Anybody wants to add something about the training programs and the challenges that you had there for those of us who had the opportunity of working with students that do not necessarily come from the right background? Okay, so we can continue to. Okay.
Actually,
yes, go ahead.
It's an interesting question about educating in
Is it not on? Well, I think so.
Okay. Oh, sorry. My name's Daniel Burkhart. I'm a scientist at Celerity. I think it's interesting here because I was just thinking about the difference between educating, say, like flow cytometry analysis. I don't think that that's something that we're trying to scale in the way that we are single cell analysis or like light sheet microscopy analysis. Just access to the raw machines and the data is so fit for purpose that people aren't doing that. And so it's kind of interesting because computational biology and single cell analysis really is a problem of statistics. It's a problem of software. It's a problem of access to computation. And so it's kind of a unique problem that we face, actually, that you even have like groups trying to build these notebooks to get, people who don't have the sort of background skills or maybe are from a very different scientific background to work on the data. So it's an interesting problem. So I'm not sure if there are other scientific disciplines that we're trying to do broad public education on like this in such a technical field.
I think there is kind of two very different ways to work with the data. One is to recapitulate the same type of analysis that the kind of large-scale data producers, consortia, etc., are doing. And that indeed needs a lot of compute. But at the same time, we've seen with genomics data how much power there lies of individuals that don't even have the intent to recapitulate these large-scale analysis, to look at their particular gene and their particular context. And it seems that there we are still failing a bit to deliver that. And perhaps that is the more impactful eventually, because the big centers and data producers will always be in a hurry. And they will miss out on so many interesting things that a well-trained person with a biological understanding, or even a not so well-trained person with just a lot of time to look at data, can find real gems that are overlooked.
This point is very interesting. And think on this, how can we bridge the gap between biologists and bioinformaticians in order to, have a conversation where everybody, are telling the same language?
Raju Plai from Los Angeles. So I want to echo the point that he made. I think data analysis is going to be a bottleneck for many biologists like myself. Even if you're able to generate data, if there are no biologist-friendly tools, it will delay, what we can produce and share. So I think one example would be you know, in the NGS field, it was similar a few years ago. And then there were attempts made to make it simple for biologists to use. Like the two examples I can think of is Galaxy and Terra. That did speed up a lot of things. I mean, if you didn't have to run your code in a command line environment, which a typical biologist is probably not trained for, versus, you know, you have an interface that makes it, you know, more reasonable to do these things. I think Jupyter notebooks that he mentioned, you know, it's a great start. You know, I mean, with some training, biologists would be able to do it. But I think the community should, I mean, we have so many fantastic, you know, computer science people in this community. So it shouldn't be a great lift to make some tools that biologists everywhere can use.
So I think that in theory, so we talked about again, building communities here, right? And what the community can do for us, each one from their own perspective, right? And we see that there can be a synergy here, right? Even between people that sit here in this room, come from different backgrounds, they can do different things and they can synergize, they can help each other. But there are still obstacles, right? And I can see it myself, right? I'm somebody who works on it, right? On the computational side. And so I want to push on this point a little bit more because I think it's important. And not all the stories are success stories. Sometimes there are beautiful tools that are not being used, and vice versa. Sometimes there are needs for types of analysis that require a certain approach that it's not being applied, right? So so the question is, what do you think as a community or as community effort or as some community force that we can envision here? What can help reduce that or facilitate a better communication? It's a difficult question, I know, and there's probably not an answer for this, but just thoughts.
I was about this point earlier. I'm Paulo, and I'm from the Boston Shipper and I want to say like about training. There is an enormous amount of like pipelines. It's like fantastic. But one of the things that I think if you want to train a person, you have to do is like let them know when it's a data set is like there's single cell RNA, seq spatial, it's bad. What they have to look for, distinguish between good and bad. Before diving into the biology, I want to know what are the features like, what are the things that I have to look into for like spending the time looking at the biology. This is not trivial because like every different modality is going to have a different like artifacts. There is no artifact-free technology. Now, if you're looking at single cell RNA, seq, you know, if you're looking at like spatial, and different spatial technologies are going to have like their own artifacts, okay? Because they are, some are like visualization and image based, other are capture based. You have to know that, and you have to know what to look for when you are like in space. I know all data sets are equal, guys. And the other thing is that accessibility. One of the things like for me is like really hard is there's still not even like the basic standard on what to upload like for the rest of the planet, in the sense that even on Geo, you will find datasets that have no annotation, or the datasets that are like halfway through analyzed, and some of them are like completely raw, but it's essentially a jungle, and that requires you to dig a lot more, understand like it's worth my time and so on and so forth. So there are a lot of like legal barriers.
Thank you. No, I think this is a great point. Just as a way of facilitating the conversation, have some basic idea of QC, but in a standardized way, so we know that we are, in a sense, barking in the right tree, right? Investing where we need to be, for sure. I think that's a great point. Other points about that? Yeah. So maybe from the from people that come from the computational side, right? When you think about your next algorithm you'll be developing, or how you are making it, how you're selling your product in a sense, and how do you have people understand what you've done and use it. So I don't know if all of the experiences that people had here in the audience were completely positive, or you had some points that you thought, I wish there were a better mechanism to make my work more clear to people. Yes.
Hi, I'm Jayanne Saunders from Vanderbilt. I'm one of the coordinators for the Pancreas Bionetworks. If anybody's interested in the pancreas, please come talk to me. I think that, so two things. One, it seems like there should be some mechanism through HCA of like matchmaking between computational, workflows and pipelines and the biologists' questions that they're asking, just because there's tons of tools out there and I can go to a GitHub repository and like, I have no idea how to judge that. So I'm going to just kind of click around until I find something that looks interesting, but there's definitely a better way to do that. And then two, I think we kind of need to invest time in establishing a shared vocabulary for what we are communicating about. So whether that's data set integration, the word integration means a vastly different things if you ask a biologist versus a bioinformatician. So I know that's like a very exciting kind of like milestone, but like I think it's necessary in order for people to communicate about goals and shared goals of the movement in general.
A dictionary would be a very good idea.
I like the matchmaking too. I think that's in a sense because there's always good to have a conversation, right? And actually talk to or at least email with the person rather than a GitHub page. Yeah.
Okay, maybe we can, does someone want to contribute also in this question? Maybe we can move to the next one. So as we were saying, there are many tools over there, but just a few of them are sometimes user-friendly or, have this API. So what do you think as a community we can do to really facilitate the use of those tools and also to plan the development of the next tools?
So what's the kind of makes tools succeed and fail is often the user interface and kind of the mentality or the design of the process, how problems are being solved. And to some degree, that's trial and error. And often it's not clear upfront which tools succeed and which tools fail. At the same time, so it definitely deserves a few tries. At the same time, there is a lot of infrastructure underneath that pretty much everyone needs. So what I would argue for that those people who build centralized infrastructure, that is kind of data ingest, data processing, these type of things, and try to, as much as possible, create API access very much upfront, such that other people can build kind of user interfaces relatively lightweight on top of the core infrastructure, so that not everything kind of obviously that undermines kind of their monopoly on defining the user interface side. But we know that user interfaces are a bit hit and miss, so it'll give many people the chance to build on the infrastructure with API access.
I think he raised it first, and then I go please.
Yeah, thank you. So, hi everyone, I'm Giovanni Palla, PhD in Munich and a core member of SCVerse. SCVerse is a recent open source project that we started with a bunch of people also in the room, which aims at being an open source project that aims at standardizing some data infrastructure and tooling for single cell analysis in Python. So let's say our goal, long-term goal, is to make something that what Bioconductor built across many years in the Python community. And I just want to sort of advertise or share that we thought quite a lot in the last year on how to improve on the development of tools. How can we improve the development process of tools and
released what we call a quick cutter template, so a repository that can be used as a skeleton for new tools that uses the scan by and undate ecosystems that should have a lot of boilerplate code and infrastructure that is useful for developers to make it easier essentially to develop new methods.
Hi everyone, my name is Romain Lopez and I'm a postdoc at Genentech at Stanford. I kind of want to raise, I think I'm also really excited about the idea of infrastructure. I'm a computer scientist and I worked on single cell variational inference and other algorithms that are part of SCVerse. And I kind of want to say that infrastructure is great, but like it's also interesting to think about the incentives for everybody to build this infrastructure. And I think that the incentives are not quite completely aligned in a sense, because so the incentives, I guess, for computational people is to build algorithms that are being used. So to do that, you need an API and you need an infrastructure. But I guess our incentive is also to publish methods papers somehow. And the misalignment of that can just be to create many GitHub, many packages, right, that never get used, but they get published. And so what SCVerse as an initiative and all of this open source software is doing is that enables people to kind of be part of a universe and to have less work to do on the infrastructure because it's already being built. But it still remains difficult because there is no mechanisms for like funding initiatives. But if you want to build infrastructure, what you really need are engineers. So the same way then, and I don't know how it works in the human cell atlas, but the same way then there are mechanisms for funding research in the lab and paying for reagents. We should think about software engineers as some kind of part of that system as well. And that's the only way to build a real infrastructure. Because at the end of the day, everybody's in academia, and maybe some people came from software engineering, some people are really talented. I'm not a software engineer, even though I wrote some good amount of code. And I can say that, and I'm kind of curious if there is this kind of mechanism. So I know that I see like, some of the software and the SCVerse, etc., receive funding from the Chance Coburg initiative, and that's enabled to have this engineering effort for keeping some of the algorithms going. And this Nir uses a lot of his personal research money for investing in software as well. But is it just interesting to reflect from the perspective, perhaps? I'm curious to see if anybody has any thoughts.
I'm sure I'm interested to hear both of you from Ruma and from Gio on how important it is that the engineers making the software usable are in the same institution as the research scientists who develop the algorithm. I don't know, like in SCVerse, I imagine there's a lot of software that's maintained by completely different people now because grad students have gone on or something like that. Is there any thoughts about that?
If it was truly open source, it wouldn't need to. Right?
But is it
that's where we are. What we're doing, is it open source or is it just within a lab, or like what are we doing exactly? Yeah,
so here you're going to the question of incentive and motivation, right? So why would you do that, right? Even if it's open source, even if it's tangible, what do you have to gain from it? And I think this is really a critical point, because in academia, the incentive system is different from what you would get in, I don't know, if you just go to the AI growing industry out there, right? It's completely a different set of incentives. And I really think it's, at the end of the day, it's all about the science and what we can do with those things and discoveries that are being made. It's just what I think, but I'm very curious to hear from, but maybe just before that, I know that you want to speak, and then I'll.
I guess it was more of a statement. I'm Nikki with the Kidney Tissue Atlas, KPMP. We have a small group of software developers, and they've actually worked to develop our tools that we currently have on the Atlas, and what allows us to expose our data and everything. And we also go through an extensive user interview process that allows us to have developed what we have so far. So we have it based on certain personas. So we have, I believe, 6 personas right now that we've developed, that we kind of keep those personas in mind as we develop things for our atlas. It's really been very helpful, and as we continue to develop, that's what we do. So
I'm curious.
we would love to have more people outside of our consortium to talk to too. So
No, this is really intriguing. So when you say personas, you mean like a user profile in a sense?
yes.
Can you give an example for one?
So bioinformatician would be one, right? Clinical researcher might be one.
Yeah.
So these are some of our personas. Like if you go to kpmp.org, you can go to software development and you can see our personas, like they're listed right there for you to go ahead and look at.
Yeah.
So
So maybe I want to pause here just for a second. Let's do a short survey, which maybe I should have started with. But can just from show of hands, who here comes from? You can define yourself as coming from the biology perspective, running experiments, really thinking about that perspective. Okay. And then let's say from the other extreme of the computer scientist and the method developer. Okay, and hybrid, somebody who thinks they do both. Yeah, okay. Okay, so we have a good mixture here. So that's, I just want to see that we have this conversation, because I think it's, we've heard right from people from that come from the side, from the biology side. And how do you see that synergism, and what do you think is missing to form, let's say, a more productive community here? So are there any more thoughts on that angle? Yes.
Hi, my name is Kaylee Matthews. I'm a science writer at the Broad Institute, and I'm on the data portal team. As someone who recently came from doing bench work to now a more technical role, I do documentation for the Broad for our pipelines for Terra. I think one thing to consider, and I think is often overlooked, is that a complicated tool can become user-friendly with good documentation. It's often overlooked because it's not really the fun part. The fun part is actually doing the work and getting those findings. But when you have good documentation, someone like myself, who was a research scientist, can start to use these tools much more quickly and much more independently if you have good documentation.
I kind of agree, but I also disagree. I think documentation will just not happen. What people need, and documentation, if anyone has ever looked into the documentation of commercial software packages, they say the obvious and not very useful stuff, and you still have to buy the book, How to XYZ for Dummies or something like that. So there, I think what we really need is the out-of-the-box examples, like where you kind of start the program and within 1 minute you have your first kind of experience of success, like some kind of a single command statement. Okay, show me an example, run me something. And not only one, like one that really has to work, give you some level of satisfaction within a minute, because otherwise people would just give up. And then not only have, I think most tools have this now, not all the tools, but then I would like to see much more like a tree, no, not a tree, like a ladder of increasingly sophisticated use cases. And if you have kind of 6 explained use cases, that's already the structure that you need to do your own stuff, kind of along this ladder of increased complexity. And that's also more fun to do than writing a documentation.
I would argue that that's part of good.
Hi, my name is Maria. I'm a research associate here at U of T, but I'm also co-founding the Virtual Human Development Consortium. And we all have the same kind of problems. We're bridging theorists and experimentalists and engineers. But I kind of want to talk about the documentation. Maybe one idea could be a running documentation where it's like a wiki page and people can use it and add their experiences to it. The other thing is, I know that there's a single cell community here at U of T. I think it's also international. So also have access to a community where you can talk to live could also be a key thing as opposed to just a static thing. That could be something you can establish.
Yeah, perfect. If I can add on that, I think the great success of Bioconductor was the vignettes, right? They were very, very well documented, and they helped lots of biologists to really go through the pipeline.
Okay, maybe just to one last, I'm really curious. We talked about, okay, what can the community do for me in a sense, right, to accelerate what they do. And so we've heard about how to use the tools, right? How can those be useful? Now let's flip it for a second. Think about if you are a tool developer and that's what you do and that's where your passion is. What do you think are the holes right now? What can be done better so you can actually be more productive, more impactful, and do things that are actually useful? Any thoughts about that? Yes.
I just wanted to say that maybe feeling that you have to always restart from scratch or not knowing where to find yourself if we can like overcome that first step, you can be effective. So again, talking, figuring out who did what and moving on, because really science is built on other people's work. So always having to reinvent the wheel is a hard thing, and knowing that others have done something and you can move forward. So I guess some community site that says, what has been done, where you can go, maybe you can ask your questions there could be a nice way to start. So you don't have to start from scratch. I think that helps overcome these first challenges.
Hi, my name is Amin Ahmad. I'm from McGill University and Mila. So my perspective regarding that question is partially is basically what something like HCA is trying to do, which is kind of data integration and having a more homogeneous and consistent set of labels and data that, each of them may be small scale, but when you want to talk about, let's say, deep learning model development or some of those models that are very data hungry, being able to have these large data sets that may come from different labs, but they have been pre-processed that, a lot of these kind of cleaning of the data and pre-processing right now is maybe more than 50% of the time that we put in to even get started with method development, because we want to do it right. But if that step is reduced, definitely people who are less versed in the biology and bioinformatics side of things are more computer scientists and deep learning or machine learning people can, basically get involved quicker.
So just to intervene here, so in terms of current efforts, right, for discussion and sharing, just like what you said, I don't know if you want to add some things here. I know that there are some efforts for that from SCVerse. Is there just for people here to, if you want to talk about it a little bit? Yeah.
On the method development side, I guess from the method developers, I very much share the infrastructure, in particular, I guess the data format aspect. I think being able to just read the data in a format that the developer knows where all the information is stored, I think has a lot of value. I think anyone in the room who has been going through data curation efforts knows very well that often in journals, data is not published in a standard way, right? You can find any kind of formats for any kind of data. So yeah, I think maybe on this question is like, I wonder what could be the incentive for having a standardized data format for data sharing, for instance, and whether the HCA could come up with some of those incentives. But yeah.
Please.
I just want to mention an answer in response to your question about, I guess, useful software. So I like the idea about personas, and people mention use cases. We develop a lot of different software like Cytoscape, for instance, not necessarily just in human, in single cell genomics, but we found it's very important to strategically identify the the persona that you're working towards. So we have some software that's just for new users, and we have other software that's for data scientists. And we know that, and then we interview those users. Either we know about them because they're in our own group, but we also go through these interview processes following the human-computer interaction field, which or usability, user experience design field. We're interviewing users around the software and doing a bunch of user testing, make sure that the software is addressing their actual needs, and not just any users, but the users that are strategically identified by yourself as the target audience. Like when we're building a new a website for non-computational biologists, I don't interview people in my own lab who are computational biologists. I ask them for advice about things, but we make sure to mostly focus on new people that we've never met. So that's been very useful for identifying, like figuring out what the real problems are in the software, which we don't see ourselves usually.
So maybe picking on that, and again, it is something are there any forums that people here in the audience had a good experience with, right? Like any chat group that people can post. I know that we are running one for SCBI tools, but if people here want to share any experience that was either good or bad in trying to interact with a medium like that. Any stories? Yes.
I was going to say, Gary, here at Toronto, the single cell analysis community, they're running I guess a Slack channel forum, and they also have documentation. Yes. And you can ask dry lab experimentalist questions or even share papers. I mean, I think Gary, you can tell them more about that, but I found it really useful. So I am a non-bioinformatician, a non-experimentalist. That's why I didn't raise my hand. I'm more of a modeler. So being able to access some of these tools is really challenging. And so I found that forum really handy because I didn't have to start from scratch.
Yeah, fantastic. I think these are, I mean, that sounds like something that is quite useful, I guess, facilitating communication, right? Either through Slack or Discourse or platforms like that, for sure. So,
I don't know if it is good or bad, but they are not centralized, right? So there are many channels in Slack that you can access. So
yeah, but there are centralized options though, right? There is a bioconductor Slack that is very active actually, and many questions can be asked on like many different topics, both I think from like an analysis point of view, then a method development point of view. And for SCVerse, we have a Zulip, which is like an open source, free mostly. That's why we chose it. Platform for async communication. And I think this kind of async communication is very, very useful, and it's orthogonal to a GitHub issue or like a discourse post where you would have, maybe like your PyTorch discourse, if anyone you know this platform, because in that case it's very much like question answering, whereas on Zulip and Slack you can have conversations that are very, very valuable, I think.
Hi, so my name is Shamini, and I'm a postdoctoral fellow here. So were you talking about panoramics? Okay, so yeah, so we have a group here called Panoramics, and it's a spatial and single cell platform. So I drive it, and over the course of the months, I realized that it's really not about advocating for computational science or computational biology, or if you're a software developer or if you're a wet lab person. It's really about understanding that it takes all kinds of people to do science. And I realized that if you actually allow people to talk and to engage and to share what is it that they want, you don't even have to do much. You just need to spark, and you just need to be that spark, and all those collaborations and knowledge sharing just comes through, because that's what you need. So I'm like a hybrid. I started out as a wet lab biologist, and I used to think like computer biology was like this domain that is like I don't know, it's like heaven or something like that. And then once I became a started to do a lot of computational biology, I realized that there were lots of computational biologists who needed help from wet lab biology. So it's you need to bring out the problems. I think we are all scientists here. And once you start to bring out the problems, these are the challenges. What can we do to make it better? People just come forward. Like a few weeks ago, I had a workshop done, and there were a group of people, wet lab biologists, who actually gave feedback that they did not want to try the software at all. And we had that workshop, and later on they were like, "Oh, thank you so much for doing this. Can we make some suggestions?" So it comes automatic. Yeah. So you have to. It's because there's a lack of understanding, and sometimes, unfortunately, there's a lack of respect as well. And it kind of goes back to what Gary just said, and also the personification that she mentioned as well. You know, it's because it's an identity, right? If your identity is not respected, who's going to come forward and share their wisdom? No one is. So I think if you can focus on that interpersonal aspect, I think the science will definitely grow.
Those are fantastic points. Can I ask you one more question? So you talked about success stories here that are it's local, right, to this area. Can you, or other people here in the audience can speculate of how you scale it up to the more, let's say, almost international level, global level of the Universal Atlas as an, I mean, not for the whole thing because it's very distributed, but something that is more distributed?
Well, it's a habit. So as you grow, so I think as you, so it's always a trade-off. You cannot have perfection. Like every organization will have its problems. Every organization will have its good and bad leaders, but it's about inculcating a habit. So just to give an example, Starbucks is doing well not because of their coffee, but because they developed strong interpersonal training programs for their staff, and that's why people go. So as long as you need to have a blueprint. And we also have to be mindful that not everyone is going to embrace this. There will always be researchers who don't want to learn. We just have to let them go, right? So if we keep having that infrastructure and we have that, I don't want to say documentation, but sort of like a roadmap or a goal where we can identify it. Everyone wants to identify with something, right? Like phenomics is about having a vision. So HCA is about building this human cell atlas. And even for non-scientists, if I tell them, they go wow, because that is like a vision that you can look up to. So you want to spread it, then you need to have that vision, that goal, and you need to have leaders who can embody that habit and promote it as you go along, as you grow.
Thank you.
Does someone else would like to comment more on this subject, or should we move to the next question? Could be. Okay, so what are strategies that can be implemented to promote collaborations and knowledge sharing within the scientific community? We kind of touched on this subject, but does someone want to elaborate more?
I can only tell you about from our point of view. So right now with the virtual human development, there's experimentalists, engineers, tools, individuals that make microscopes, and also modelers. And we all talk different languages, although we're all asking the same questions, we don't have the same languages. So the first thing we're doing right now is having discussions. And I think you mentioned this once, the matchmaking. So we're already doing that, where we have one theoretician and one experimentalist, and they both meet for the same topic, but they talk about it from their perspective. And then there's a good 45 minutes of discussion. So the key is discussion, not just presentation. And at least from what I find is the community really appreciates that. And what happens is there's an exchange of ideas and also definitions. So once you're able to understand the other side, then you can actually interact a bit better and collaborate. So I think that's actually one
main point is being able to understand each other, right? Oh, can I just say one thing? This persona, can you make a persona of an individual who does not like to download a lot of things or feel like it's like the 1980s and then I have to figure out, or something that's version control?
We're working on.
Yeah, that's a persona that I'd like to.
I love that.
More comments on that?
Should we move to the last one?
Yes. Yeah, so we also would like to touch based on challenge associated with researchers in resource limited settings. So in this case, what would be those challenges and how could we address those challenges? For instance, I'm from Brazil, and to really run an experiment there, I have to import all the reagents, and they are 5 times more expensive than here. So and take longer, super long.
Can't help with the wet lab part, but again,
only related to wet lab.
I know that's why I'm just jumping in and switching it to the data part. One of the other things that we're looking into right now are creating what we call data enclaves. So the enclave would then allow a user to come in and use our data and have compute space, right? So if they don't have that compute space where they're at, right, it's in the cloud there. I mean, we're still going through it. There'd be data use agreements, right, depending on what the data is and all that. But that's something that we're looking into because we understand not everyone has the same resources.
Hi, I am Daniela. I am from Argentina, and so this topic it's really important for ourselves, and we have different problems. One is data generation. Yes, I think there's no one in Argentina that generates data, and most of us are computational biologists. Myself, I come from a physics background, some kind of modeler too. And we kind of have to do all the things because we don't have a developer on the computational biologist and the bioinformaticians. So we have to do everything. And well, that's kind of a problem. And I just don't have any solution. But I just wanted to give my perspective.
Yeah, I think hearing about the problems is also very informative, right?
Yes.
Not just solutions.
I don't know who raised first, but
I would have a question for what you just said, but what also some other people implied. I sometimes, so we're producing a lot of data and analyzing the data, and that creates a situation where the data production drives everyone into stress, basically. So data's coming, the data needs to go into the first paper, and then already the next data set. So I feel like we do not look deep enough at the data. And sometimes I remember the days when I was a PhD student when we were not generating data and we were just analyzing data. And I felt we had more depth of the computational, like more depth of looking at the data. So to what degree, kind of what are the obstacles of, especially for those people who are neither data producers nor methods developers, but biologists of a theoretical or small-scale experimental nature? What are the real obstacles to use that time that is not going into methods development or just riding the wave of massive data coming out of the lab? What are the obstacles to use that for a much deeper insight into the data than the primary data producers can achieve?
Does someone would like to answer? Yeah.
Hey, can you hear me? Okay. I'm Brian Averman from CZI. So I think the problem or the limiting reagent over there is going to be metadata, because pulling data down publicly and trying to spend time with it, the first thing you're going to run into is, I don't know why these data artifacts are in there, why these residuals, why this, why that, right? So having a good schema, having good metadata collection routine, all that kind of thing, very important for reuse. So.
Yes.
Can you hear me? I believe this is something very important to follow the FAIR principles when depositing the data. And another that I have already to deal with, I submitted some papers that we reanalyzed a lot the data to Giga Science. That's a journal that in theory accepts the reanalysis. And the editor said, you're not generating any data. You are only reanalyzing data. So it was refused at the beginning.
I had the same experience.
It happens a lot. Yeah.
Yeah.
So the culture of the journals.
I'm sorry. Oh, you can go first. Yeah.
Hi, I'm Sergio Triana. I'm a postdoc at the Broad Institute. I'm also been co-organizing the Human Cell Atlas workshops, virtual and in person, over the last couple of years, which is kind of related to this question of resource limited settings, and also to some of the other questions that we had so far, where you, like, I think the question of scientific community and how to engage, it's quite context dependent. It's very different to approach a community of someone that is already part of the Human Cell Atlas and is already working on single cell, to when you're trying to train or spread the word of single cell and Human Cell Atlas to people that are scientists, but not necessarily in the field. I think the limitations are very different. And I think that's kind of the case in a lot of places where resources are limited. The exposure that you have to Human Cell Atlas and to these kind of technologies is more restricted than the one that you have in the US or in Europe. So not only it's a question of, well, funding and resources, which is quite limited, but also of accessibility to the information and the knowledge. I think we're trying to do that with the workshop, but I think it's also, even when you're defining this workshop, it's very specific to the place. Like as the previous one of the person was mentioning, a lot of these places have a very strong computational background because a lot of the stuff that they do is bioinformatics. So that means that if you're going to do a computational training, it's actually relatively advanced and quite advanced because people are quite trained on this. But if you're going to do wet lab, you kind of have to adapt it so it's low. You teach things that are cost effective or things that are actually feasible. It doesn't make sense to go and teach someone how to do CRISPR screening on 10x genomics in a place where that will be the funding for the whole year. So it kind of really depends on the community that you're trying to include and what accessibility you want to have. The same thing goes to a lot of the questions that you have. The kind of interaction that you will have between a software developer or a wet lab person will change depending on the place that you are. In a lot of places, there's just not going to be wet lab. So you kind of have to connect computational people with computational people and wet lab people across the world, or that are willing to share data with another person. So I don't have an answer also for these questions, but I do think that when you're thinking about these questions, it really is context dependent, and it really depends who's the public that you're trying to reach.
Okay. Hello, my name is Amir. I'm a PhD student in Jesse Gillis's lab here in Toronto. So this is going back to what Christopher was saying, and I don't know if it's exactly in the purview of the question as posed on the slide, but so it's my impression of the field by reading stuff is that so you were saying there's a experimental biologist and then there's a computational biologist, and in computational biologist there's like methods developer and then there's modelers. And the question is, what are the modelers doing? Somebody was saying, I come from a physics background, but we're looking to model. I think I'll give you my opinion. My opinion is that we don't have a well-defined problem space. So let's say COVID modeling as an example. We have an intuitive grasp of things. In epidemiology, disease spreads. People come into touch with each other, they cough, and then other person has a disease. That person goes somewhere and that disease spreads. So we have a sense of, okay, over time and space, people give disease to each other. So if we sort of restrict them, then it's not going to spread. And then you can come up with some math to capture that phenomenon. I think in spatial transcriptomics or single cell RNA sequencing, yes, there are some problems. Everybody's talking about integration at the conference. So, okay, so, there's like some defined problem. Then there's cell-cell communication. But I think the general, the general impression I get is it's not super clear because we don't have an intuitive grasp of the biological phenomena the way we might have of disease spread in COVID. And earlier today, there was like spirited discussion about like foundation models, right? Let's put the thing, this multi-omics stuff in the embedded space and get something out of it. And I think people are motivated by this. Let's use all of the data at the same time to have global insights about phenomena. I think that's really what's driving the field. That's my impression. So, I think to answer the question of like what to do in resource limited settings, experimentally, it's clear we need more money, for example. It's like, okay, we can't come up with a solution because the government has to come up with money or something like that. Computationally, yes, there's method developers. People in resource limited settings, if they can share computing resources, I think it was a great idea with, countries like Canada. I think that's fantastic. But then there's the problem of like the modeling situation in which what are the modelers modeling? I think that's, to me, maybe because I'm too new to the field, it's totally unclear what we're supposed to model. My diagnosis of the problem is because we don't have an intuitive grasp of the phenomena the way we might do in epidemiology, where it's like clear, genes are talking to genes in different cells. Like, what does that mean? But people show pictures like, oh, hepatocytes are like spreading. Then we get a grasp. Oh yeah, these things move and they get bigger. Great. But I think this global, insights and UMaps, in my opinion, we don't know what we're modeling. That's why, the question of like, what can the computational modeler in resource limited settings can do? I think the reason we don't know that answer is not just because it's resource limited, but because we don't know what the question is. That's like maybe a controversial thing, but that's sort of what I feel about this.
But that's a universal, I guess, conundrum of the if you want to make sense out of data. So I guess we are almost running out of time. So I just want to end it with a question here for everybody. So I started by asking, okay, what can data do for you, right? I want to think about it from your very centric perspective and see, okay, how can I gain from it? What am I missing? But let's flip it just for the last few minutes here. Can people here share seeing the community here, hearing the need and so on, what do you think from your own perspective, from your background, from your strength, what do you think you can do? I mean, of course, I'm not signing you on a piece of paper or something like that, right? But just conceptual. What do you think? Any comments on that?
Hi. So us there are actually from different data portals for the Human Cell Atlas, and we are here to take your data and take all your metadata and put it in a standardized format and make it publicly available. I'm here representing Human Cell Atlas data portal and UCSC cell browser. Do you guys want to say anything? I'm just here for the data portal.
And yes, I'm here for cell by gene and CZI more broadly, but yes, and we are also to kind of address a couple things that were said earlier about having data available to not have to download them all the time and having them versioned. We just launched the CT Census, which is making that available. So version 1 is live. So it's an API and you can just download it versioned. So we are going to be releasing every 6 months. So you no longer have to store them in your post 1980s environment. You can go back. So,
Thanks, Brian.
You're welcome.
Yeah, so come talk to us. And if you go to data.humancellatlas.org, you can access the data portal. And we also have a curated list of tools and visualization portals and software. So that might be a good place to start instead of just looking through people's GitHub repositories. I also have to do that. And it can be really frustrating, or it can be easy, depending on how good your documentation is.
And the question, is there a way for people to communicate with you directly?
Yes, you can email wrangler-team@humancellatlas.org.
And this link is on the website. You can see it there.
Yes,
yeah.
yes. Or you can just email me.
Awesome.
It's not on here, but yeah.
Yes.
I just want to answer the question, what do modelers do? So I work with Gary Bader, and we're trying to build what we would call like a digital organoid. And what models can do that right now you don't see is predicting dynamic data. So a lot of at least the data that you capture is usually static, single time points. And what we're trying to do is trying to understand gene expression across time and dynamics. So that's what the models can do.
Yeah, and that's also very important for us as a community to understand what the other person is doing, definitely. So thanks for explaining. Yeah, for sure.
The dictionary.
The dictionary. There we go. Okay, other thoughts about your own contribution to before we wrap up? That was very helpful about the portals. Thanks. Anything else?
There's someone from the Zoom
Yes.
wants to answer any
do
of
that.
the questions.
Was somebody do something? Oh, so somebody raised their hand.
I don't see. Okay.
Well, so yeah. Oh, please, let's do this and then I'll...
So for users who want to contribute spatial data, is there any accepted format of the data?
So that depends on who you're asking.
Also 23, yeah, we have something on Esiverse.
So for the data portal, we can take your raw data and we will curate your metadata so that all of the donors, samples, and protocols, et cetera, will be linked to the raw FASTQ files. So that's kind of like the purpose of the data portal, because that can be very difficult on a lot of archival services. They don't have that mechanism. And for UCSC cell browser, and for data portal, we can also take your analysis products, and those will be linked to the metadata schema with all the donor information, sample information, everything I just said. For UCSC cell browser, we take your expression products and visualize them. So that would be like your analysis products, your UMAP. We also now do attack-seq data and spatial data. So we can host like your slide on UCSC cell browser, and then for attack-seq, there will be like a peaks file that you can visualize. So that's new. Yeah, and then Brian, do you want to talk about cell by gene?
Well, what was the question again? That was about standard formats, or
Yeah, for spatial data,
for
some
spatial
of them are imaging based, and there's no FASTQ files.
Right. I mean, I do know that there are H580 versions, flavors that can support many spatial platforms, but obviously, like you're going to hit a wall eventually when you get into like an entire Murphish like petabyte of data, and then you're like, well, where do you put it? But yeah. I mean, like cell by gene, we're working on using the SOMA. Our back end for the census is the SOMA format, which is a container that lives in the cloud, and that can ostensibly extend it to support any data, right? Genome tracks, spatial. We're not doing that yet. We just launched the SCRN-seq version, but the idea is for it to be flexible enough to like start putting more things into the container so that you could API into it and get what you want.
Yeah,
we'll get there.
Yeah, sure.
I just, for anyone who hasn't seen the SOMA API or the cell census data access, like a lot of people have been talking about access to compute and internet speeds. I think this is actually one of the coolest things I've seen in single cell recently, because you can get like very precise chunks of data that you're looking for with defined metadata. Like if you haven't seen it, check it out.
So what we just had now, no, that's great. We just had a live demonstration of communication across the community, which is exactly the point, right? So we can think about it as a lesson, right? But as long as these communication channels exist, right? Exactly this kind of communication, that's what I guess this session is all about, right? And
I have just one comment.
Oh, please.
Hello, everybody. My name is Paramesh. I'm with the Tuckma group here, Princess Margaret. I'm from the computational side. It's great that we are generating all this new data from HCA, but in the context of talking about FASTQ files, for example. I just wanted to put this one comment out there for consideration. What I find, like, there is a lot of already available RNA-seq, not single cell necessarily, but maybe single cell too, in this huge archive database like GEO, for example, Gene Expression Omnibus, which is pretty much the standard platform where journals ask you to upload your data, FASTQ files, raw data, and some versions of processed data. But then I find that once it's deposited, it's almost like the people who generated the data, they forget about it, paper published and done. But later on, after several years, somebody wants to make something out of that, like reanalyze it. There doesn't seem to be a very friendly way of taking the data from what's available in a certain processed format and taking it from there and doing some more analysis on top of that. Oftentimes. So is there any initiative, or does somebody want to comment on that when it comes to making use of already existing data? Because as far as I know, there's tons there.
So that's a great point. I'm sorry, we ran out of time. We actually should have. But that's a great question. And we have the people of the data portal.
We do that. That's our
Yes,
job. So you can
exactly.
come to us.
Exactly.
We've
That
already done it
conversation
for
needs
you.
to happen. Exactly. This is great. No, that's the
We are doing the matchmaker, right?
Yes, exactly.
Yeah, so I just want to thank you all for being here today. It was a great discussion. I think we don't have the answer for many of the questions yet, but it's good that we are at least thinking about. And please, let's do this dictionary or this matchmaker, because I think they're going to facilitate a lot.
Right. Thanks, everybody.